Welcome Hyun Seok, who will speak on mouse fMRI with optogenetic silencing, reveals neural interactions, underlying resting state fMRI. On to you, Hyun Seok.
Thank you for the introduction, and I'm going to share my research on mouse fMRI with optogenetic stimulation to figure out the underlying mechanism of resting state fMRI. So the functional connectivity is basically a measurement of spontaneous neural fluctuation, and it defines a connection strength between regions based on the correlation coefficient. And this approach has contributed to reveal functional networks or build a connectome in the human brain. However, despite this prevalence in human neuroimaging and neuroscience field, we are still lacking the clear understanding of its causal mechanism. While it is often assumed that it represents the neural interaction between brain regions, which is connected through structural connectivity, but many studies so far has shown that structural connectivity only partially explains the functional connectivity, and also they suggest other contributors to functional connectivity, such as neuromodulating inputs or non-neural based motions, and the existence of these other confounders, they obscure the interpretation of functional connectivity results, both in normal and pathological state. So here we raise a fundamental question that whether functional connectivity actually reflects the neural interactions or not. So to address these questions, so here's an example of the human brain, and let's say these two bilateral regions are functionally connected, and if this actually represent the causal influence between these two regions, then if we manipulate the neural activity on the one side, the other side would have the response to it. And we are trying to answer this question by mouse fMRI for reasons. First, there are a variety of tools to manipulate the neural activity in genetically specified neurons. So it's going to be, they provide very high cell type specificity and also spatiotemporal resolution. And the other thing is the human functional networks are phylogenetically conserved in mice, which makes the mouse an excellent model to test theories and hypotheses of neural mechanism of fMRI. And in this presentation, I'm only focusing on the optogenetic fMRI results. And here we begin with characterizing the difference between structural and functional connectivity. And based on this, our own data and the literature, we were able to characterize the functional connectivity as its bilateral or mirrored connectivity between two hemispheres, and also widespread connectivity, assuming the neural activity is propagated through multiple synapses. So we first aim to see whether optogenetic stimulation of these cortical regions evoked a similar response pattern with functional connectivity. So for this, we developed this integrated optical MRI imaging system for flexible stimulation throughout the cortex. And after co-registration of mouse brain atlas and the images in the native space, we were able to stimulate the cortical regions sequentially in the single subject. And for better specificity, we acquired CBV-weighted fMRI from the 24 transgenic mice, and all of these experiments was conducted under dexmedetomidine and isoflurane anesthesia, which is most commonly used in mouse resting state fMRI. And we targeted these 6 cortical regions for stimulation. And here's the result by optogenetic activation. And surprisingly, the response by optogenetic stimulation was very difficult from what we have seen in the functional connectivity, as they are predominantly ipsilateral responses, while mostly constrained by structural connectivity. For further analysis, we quantified the response ROI-based, and we focus on 2 indices here. First one is bilateral symmetry of the response, and as you can see in the scatter plot, the symmetry by optogenetic activation was much weaker than functional connectivity, and also these responses were only weakly correlated with functional connectivity. which suggests functional connectivity may not represent evoked neural activity, but something else. And then why this would happen here? And we thought there would be some conceptual discrepancy between optogenetic activation and resting state fMRI. Well, in this optogenetic activation, we measure the strength of evoked activity and its propagation to other brain regions, which is kind of different from the resting state fMRI, which sees the spontaneously and internally generated interactions. So we came up with an alternative approach, the optogenetic silencing. So in this case, we specifically stimulate inhibitory neuron only, and in turn, they suppressed very strongly the neural population activity. So as you can see here, the spontaneous activities is just silenced during the stimulation of inhibitory neurons. And this is an example. So the previous study has demonstrated this approach is able to measure the elevated spontaneous activity in ACC and its connections to other pain-related regions in a chronic pain mouse model. So given that the optogenetic silencing can measure the strength of spontaneous neural interaction, we suppose that it's going to be more conceptually associated with resting state connectivity. And this is the result. So you can see upon the stimulation of 6 cortical regions, we observed the negative fMRI responses by the reduction of spontaneous activity. And we quantified this negative response as a spontaneous connection strength from the stimulus region. And surprisingly, the optogenetic silencing induced the connectivity very different from optogenetic activation, which indicates the difference between evoked neural activity and spontaneous neural activity. And through the same procedure as before, we quantified the responses in ROIs, and we found optogenetic silencing induced strong bilateral responses. As you can see here, the bilateral symmetry is almost comparable to functional connectivity. And also, these responses were strongly correlated with functional connectivity throughout the cortex. So this result suggests spontaneous neural activity might be the source of functional connectivity. So far, we have observed this evoked neural interaction resembles the pattern of structural connectivity, and spontaneous neural interaction resembles the pattern of functional connectivity. So we set our next goal to see whether we can explain these optogenetic fMRI responses by polysynaptic structural connectivity. So we began with this whole brain voxel level structural connectivity from previous publication. So basically, this matrix describes the connection between 15,000 pulses throughout the brain. And by simply multiplying this matrix, so the power of this matrix, we obtained this nth order structural connectivity. And here I took an example of primary visual cortex. And as you can see, with the increasing order of F structural connectivity, the connectivity patterns is getting broader and bilaterally symmetrical. And finally, we converted this matrix into the ROI level. So now the matrix has connections from 6 source region, which is same as the optogenetic stimulus region, to target ROIs. So first, we have observed the strong linear correlation between monosynaptic and optogenetic fMRI, while the optogenetic activation is more highly correlated with monosynaptic connectivity. And for the second order polysynaptic connectivity, we calculate the partial correlation coefficient while regressing out the first order, because these polysynaptic matrices have very strong interdependency between each other. So we want to see the unique contribution of this specific order of connectivity. And as you can see here, there is a still significant correlation with optogenetic silencing, but not with optogenetic activation. And we did the same procedure for third and so on. And finally, we did a statistical test of this value against the null distribution, and it showed us that up to third order structural connectivity might contribute to the opto-silencing responses, but not for opto-activation responses. And put together, this suggests spontaneous neural activity may propagate polysynaptically, while evoked neural activity does not. So to summarize, we began this project by characterizing the functional connectivity, which features bilateral and widespread connectivity, which is not explained by structural connectivity. And also this property was not explained by evoked neural activation, by optogenetic activation. And next, we tried optogenetic silencing, and it revealed the strong bilateral and polysynaptic properties of spontaneous neural interaction, and we believe it underlies functional connectivity. So I would like to thank to my colleagues and thank you for listening. And for details, please visit my poster session on Wednesday and Thursday. Thank you.
Yep, I think we have a question here.
Hello, very cool work.
Thank you.
I was wondering, in the activation, basically I'm doing the same experiment but with wide field calcium imaging instead of fMRI, and I observe a clear, even if I activate, I observe a clear inactivation of the areas, which is explained, I mean, my explanation is that there's an overall, you know, even if you are expressing the activatory, the excitatory opsin and you send light, basically you're recruiting a lot of lateral inhibitory connection that basically stabilize the network and make the network go down, even if you are basically stimulating the excitatory neuron. So I was wondering, did you observe, because I think your images are p-values, right? They're not the, or when it was red, it was because you had a higher activation? Or it
The red means the positive response, so
Okay.
it's basically the increase of CBV.
Okay. So basically you have, but that's fMRI. So maybe the explanation is that you observe, at the fMRI time scale, you still observe an activation because you are still increasing the overall activity, even if you observe it at a faster time scale, that activity is inhibitory. Maybe that's it.
Yeah.
I'm thinking out loud, yeah. Yeah, maybe, yeah, this makes sense, yes. Maybe we can, sorry. Yeah. I think at a faster time scale, I'm quite sure, and it's quite consistent, there's also a paper in Neuron, that if you send even an excitatory stimulus, you will basically see an overall inhibition of the area, which is, I think, a stabilizing effect of the network. Probably, if you see at the time scale of fMRI, you lose this because you are just looking at the overall neural activity, which is increasing even if the actual neurons which are working are inhibitory neurons.
Yeah.
Something like that.
That's a good point, but why we see the positive fMRI response is due to the breakdown of EI balance, where the excitatory component rises compared to the inhibitory components. So I don't, yeah, maybe actually we can see the inhibitory neural activity if we push the temporal resolution very low, very like high. But I don't have the answer to your question for now.
Any final questions for Hyun Seok? Yep, we got one more. Perhaps we'll make this the final question, given that we're well past the hour now.
Thank you very much for the impressive presentation. Have you checked the neuronal activity after optogenetic activation or interneuron inhibition using silicon probe or some electrophysiological? And did you observe any differences between optogenetic activation and optogenetic, how can I say, inhibition induced?
Yeah, of course, when we give optogenetic activation, we see there's a surge of neural activity during the period of the stimulation. But as I showed you before, when we give the optogenetic inhibition, silencing, the population activity is just silenced during the stimulation.
maybe I've misunderstood. So you activated inhibitory neurons.
Yeah, that's what I mean by the optogenetic silencing. But
I see.
but the proportion of inhibitory neuron is sparser than excitatory neurons. So when you look at the population activity, it's overall decrease.
I see. Okay, thank you.
I think that takes us to the end of our brain stimulation session. I'd like to thank Hyun Seok, our final speaker, as well as all of our other speakers. Please join me in thanking them. And thank you very much for attending the session and staying on till well past 7 p.m.